Alzheimer's disease is characterized by the accumulation of amyloid beta plaques and hyperphosphorylated tau protein in the brain, leading to the formation of neurofibrillary tangles that aggregate inside the neuron and induce cell death. Neuroinflammatory reactions in the brain are a key factor in the progression of Alzheimer's disease, and microglia and astrocytes are the primary cells involved in the neuroinflammatory response. Microglia are the resident immune cells in the central nervous system. They become activated when they are exposed to a threat to the CNS, such as amyloid beta, which they identify with damage-associated molecular pattern, or DAMP, sensing receptors, including toll-like receptors, scavenger receptors like CD36, RAGE, TREM2, and others. Early in AD pathogenesis, the mounted immune response by activated microglia exerts positive effects like the phagocytosis of amyloid beta. However, prolonged activation of the immune response results in reactive microglia and astrocytes that lose their homeostatic balance and release excessive amounts of toxic products, including reactive oxygen species, nitric oxide, pro-inflammatory cytokines such as IL-1 beta, IL-8, and tumor necrosis factor, which damage the blood-brain barrier and induce tau accumulation and neuronal death. Additionally, exposure to amyloid beta can activate the nuclear factor kappa B pathway in microglia. This leads to the assembly of the NLRP3 inflammasome, which initiates a molecular signaling pathway that facilitates amyloid beta aggregation and neuronal damage. Systemic inflammation can also promote Alzheimer's disease progression by triggering the recruitment of peripheral immune cells into brain tissue through the weakened blood-brain barrier, where they can exacerbate neuronal damage by creating an inflammatory cytokine storm that further affects glial cell function and damages neurons.